Okay, welcome. Let's begin. So today I will talk about multiple alignments. Let's begin with a few announcements. June 3rd, there's no lecture because of shortage of rooms. The examen will be on July 8th. I assume in this room. Lothar, is that right?
Sorry?
The exam will be in this room?
Yes.
Yes. Okay, last week we spoke about multiple alignments. Now we're going to, about 1D alignments, 3D comparisons, and we're going to go on today about multiple alignments. Let's recap. 3D comparisons. So there's a version of 1D strings, 2D contacts or distances between residues, one residue to another residue. Diagonal is the peptide bonds, so residues close to each other. The three-dimensional structure. This is sort of an equivalent presentation. This is losing a lot of information. I talked about comparing three-dimensional structures, so finding the similarities of these domains in 3 dimension, and I talked about finding it in 2 dimension, in 1 dimension, sorry, for a single string, just looking at sequence comparisons. The guaranteed to work program, or the best program, is dynamic programming, where you essentially do a brute force through all possible solutions, but it's not fast enough. There's a hashing that does it much faster. BLAST is the major thing, and the major problem of BLAST is really the finding against the background. We cannot do a BLAST against more than a pair because the problem explodes. So we have to have a hack, the one from Rost. We do pairwise. We first do an all against all and align pairwise the most similar ones and put the other ones into that pair. So let's begin, or let's continue on that theme of multiple alignments today. PsyBlast is the first candidate, the mostly used alignment program in the world. So that's a profile. What I mean is you have a bunch of proteins. So every row here is one protein. In this particular case, they happen to be, don't worry about that, SH3. This one is from a human and this one is from chicken. Just give an idea. What the purple columns mean, that those are entirely conserved residues. So throughout this fairly divergent evolution of SH3, you find some residues almost entirely conserved. Those are the purple points here. Now, if you come in with a new sequence, you somehow, what you observe there, what residue is conserved, what is not, is very, very important to do the next one. How could you, in fact, use that then to improve your alignment? So again, last time we spoke about pairwise comparisons. Pairwise comparisons use simply the sequence of A and B and compare them. But now I have more. I have A, B, C, D, E, blah, blah, blah. I use so many in there. And now I want to bring in something new. How can I use that information that I get from this comparison?
For the new project, first we will try to align the bases that are reserved in the other proteins, and then try to align the rest of it.
So here's the idea. Let me repeat this idea. The idea is and I made a mistake here. I don't see my next slide. So getting back to the slide, if I want to bring a new residue, then first in this particular case, I have 4 entirely conserved residues. He says that first I align those 4 residues, and then I sort of fit everything else. And there you have an idea, right there. That would be an alignment program. In fact, just like that, I don't believe anybody ever tried that. And I actually wonder why. So this strikes me as a good idea. Something must go wrong. But that in principle is the idea. So again, we use a scoring matrix, or we use conservation also means something. It does not really have to be the same letter. Remember, not always the diagonal here is the highest, but it should somehow be something that relates to this profile, and the most conserved is the most important, scored in some sense by the conservation at that very point. That's the idea of PsyBlast, and PsyBlast in fact was not the first program that did it. Here are the 2 main people behind it, Stephen Altschul and David Lippmann. Webb Miller, Tom Adden, Alejandro Schäfer. Again, Stephen Altschuler here. He's a senior investigator at NCBI, mathematics, statistics, and biology, PhD at Harvard, master at Harvard in mathematics. About 70 publications, 2 over 50,000, 8 over 1,000, 120,000 citations. So this number is not very high. This is incredible. An H index of 44, again, is not so stellar, but the impact of Stephen Altschuler on the field is enormous. Another person with not too many publications, MD at State University of New York, Buffalo, master in Brown University in mathematics. He has been the director of the NCBI since '89. So the NCBI is at the National Institutes of Health, this institute that is focused on computational biology. In Europe, we have the EBI in Hingston, Cambridgeshire. But that is older, and in fact, the EBI has changed the director 3 times. Now it's Janet Thornton. Awards: US National Academy of Sciences, ICB Fellow, ICB Senior Scientist Award. H-index not that high, but immense impact on the field. The concept of PSI-BLAST is again the fast hashing, the same thing that you do for pairwise sequence alignment. In fact, that's exactly the way you begin. Then you do in dynamic programming to in fact extend these findings that you have. The dynamic programming is okay, is fast, because now we, in fact, the dynamic programming is proportional to the length, or the product of the length of the two, and since now we only have to align short regions, this is very, very fast. The problem here, again, is to compile the statistics, background signal versus the hit here. You collect all the pairs that you have and you build a profile, and now we are back to the step that we had before. This profile, you align a sequence coming in to that profile, and in fact now you do it simply by aligning into the vector. So every single position gives you a probability of having a particular residue in there, and you just compile a product of these probabilities essentially. Okay? This is in principle like the simple hack that you proposed. In fact, your hack is a faster solution. People did something like the hack by looking at consensus. I'll get back to that. Consensus meaning, well, so just read this profile and write at every single position the residue or the letter that is there most often. And then take that as the sequence. In some sense, it's what you say, but your idea is more specific to really the idea of a motif. The problem with the idea of having the conservation that you said is it's very, very dependent on alignment mistakes. So you bring one wrong thing in here, you lose the conservation in that sense, in the strict sense. Or the probability, so in the number that you have here, the probability is not changing that dramatically. Okay? I'll get back to that statement in a minute too. It's not entirely true. I'm sorry. So I take the sequence, align it into that profile. Now, what do you believe is the next step? So I have now taken the sequence, aligned it into the profile. Now what? Again, let's sort of go back to the beginning. We have a sequence that we want to align. This one here, or originally was the SH3 in human, the first one here. We search in the database. We find all the pairs, all the other ones that fit, and align them into that one. Build a profile. Okay? Now what do we do next? I can tell you that position, P is, this thing is called PSI-BLAST. P stands for position, S for specific, and position specific essentially is exactly this idea. You have a profile, you have one particular position, and you align according to the information that is specific to that position. What do you believe is the I standing for? Index. That's a great idea. That's a very good idea. So BLAST got so fast because of the way it does the indexing. So one essential aspect of BLAST is indexing. Totally correct, but it's unfortunately, BLAST was called BLAST before the PSI-BLAST. PSI-BLAST is 97, BLAST is 90. And BLAST originally had this indexing, so it's not indexing. Great idea, but that one didn't fit. Any other idea? Iteration. The idea of iteration, anybody can, now that I've sort of revealed the word, interpret the word? Well, the point is now oops, I lost my profile here. But now that you in fact have this profile, you can run again with that profile. So initially you started with just one sequence and found all of this profile. Now you're doing much better. You have a profile, you can run against the database again. Right? Build up a new profile. Now, let's look at what we can reach with this approach. Let's just assume that we have a family U. And family is defined as everything that has a similar structure in this context. It could be a similar function. But just think about it as some definition that you can name. U is the unknown protein, the search protein. You start from here. In the first step of BLAST, what you're going to do is you're going to find a circle around that sequence, right? Because you simply compile an e-value. And the e-value is based on pairwise comparisons. So that essentially is just a circle around the initial U, which is at the center of the circle. Now let's just assume that by your cutoff, I call it safe zone here, by what you believe is good to mostly get you the right alignment, you find those white things here. Then you build a profile. Now building the profile sort of shifts the focus. Because U is at the center of the circle, the original black circle here, blue circle, but most of the findings come from that side of the circle. So the profile will sort of shift the way you look into the world to that side. Simply because the profile will be dominated by things that lie here. Now you do the iteration. So you do the next step. In the next step, you will in fact widen this lens here. You have a larger search space. But what it actually means is that in the second step, you lose this part here of the original safe circle. So there was something that a pairwise BLAST could have found. It didn't find it because it was not in the database. But if someday somebody puts it back, the profile will not find that anymore. Although the profile is much more sensitive, finds many more things, it actually drops something, right? As a user of the PsyBLAST, in fact the way PsyBLAST is done, you always see at the bottom of the list the last run. In fact, it may even be true that there was a hit and it's not in the final run. But somewhere, so we're talking about files that can be 100,000 proteins long. And somewhere on the top, you may have a protein that was dropped, although it was an absolutely clear alignment, simply because the profile no longer favors it. It also is true, remember I talked about this iterative sequence search. So I have a triangle, A does not find B, B does not find C, and it also doesn't find C. But then we have an intermediate sequence. So I had this example at the end of last time where A, B, C, these circles don't, well, that's not a good one. But then I have a sequence here. I needed a slightly different drawing. My circles must be bigger. Then I find some protein here that lies within the circle. These circles have to overlap. So I find a protein in between the overlapping circles, and then I can infer that A and B have a similar structure because the X sort of connects them. And this idea is illustrated by the disc, the silver bluish disc here. So I can draw a circle around everything that I find, every white one. And these circles together, in fact, are larger than this lens. That would be another strategy. The problem again is, so one of the reasons why PsyBLAST was not the first program that in fact used profile to sequence alignments. But it was the first time that a profile to sequence alignment was put forward in a very, very simple way that was very easy to use. And the problem really is, in some sense, what you said about using only conserved residues. I said we use probabilities. Ultimately, there's a long line of possibilities in between these two extremes. Of things you can try, and that is what people tried. And they left it to the user how to use the profile method. PsyBlast didn't. Another thing is that PsyBlast is fast. Look, compiling all these circuits around these points would take a long time. So getting this profile, essentially one blast run pair, you start with one sequence, you run against the database, that's in the ballpark of 10 minutes today. No, it's in the ballpark of 2 minutes today. In this particular example, so 2 minutes would give you essentially this circle. Now you start with all of these here, you have 10 times 2. You already have 5 times that time, right? While just running with this lens is one additional run, another 2 minutes, not 5 times 2, right? So this would be great, but again, it takes time. Now you go further. Just assume that at some point, so your lens, the view on the world grows, you get more and more of this family, but let's just assume at some point you pull in the wrong thing. The moment you pull in the wrong thing in another family, that completely poisons your view here. The problem is that this is exactly in some sense also your idea. All you need to do is hack into this conservation. So essentially put in one sequence that is totally wrong, and you will substantially change the profile. Not as strong maybe as when you look only at the conserved residues, but still this will be sort of the strongest influence because divergence counts. The important thing to look at here are differences. Things where proteins are too much alike don't tell you anything. We are really caring about the divergence. So the first mistake here hits you. And that essentially is the problem with polluting PsyBlast. And there is no clear solution in how you can avoid that. Do you have any idea how you could avoid that? What could you do to avoid this pollution? How could you control for pollution? Again, pollution is defined as you iterate, and at some point you pick the wrong thing, and your iteration doesn't converge. As long as you don't pick the wrong thing, you hope that ultimately it will converge. You will just not find anything more. But how can you avoid? Throw some ideas at me.
First iterate and create a profile and save it, and then maybe 3 iterations later looking at that value again.
And?
I mean,
what do we look at? Oh, the number of. So essentially, he says we record how many hits do we have, and I assume you want to say at some point we're going to have an explosion of hits because we pull in an entire family, and that sort of gives it away. And that, in fact, is one safeguard people have been implementing. The problem with that safeguard is it may very well be true that your profile gets you into a new part of space. Well, I don't have the point in between. So the lens gets just so big and you suddenly get a view on this part here, and that may be very, very highly populated. So yes, when things go wrong, you have an explosion like that. Unfortunately, sometimes also when you find something, a new treasure or a new jewel or something that you're hunting for, it may also be an explosion. That's a little bit the downside. But this is one safeguard that has been implemented. Any other idea? Do you have an idea? So one thing that you could do, I'm just paraphrasing or misusing you. One thing that people have done is in fact put the sequences backwards into the database and looked when do they pick the backward sequences up. And backward sequences by that ratio that I explained, rationale by the current belief, don't exist, so it is a mistake, stop. that was a clear moment to stop. So that's another example for you to illustrate how much we believe that these other sequences in the other direction don't exist. And that in fact is still being used. Any other idea? So you could put things, so for every, we will get back to secondary structure prediction and the like, you could put into the database copies of things that are totally different secondary structure, or that are, for which you know they most likely will really look differently, so you're not supposed to find them. And that again is a safeguard that many people in everyday searches for being careful apply. Unfortunately also CPU intensive. Okay, let's get back to the next step. PsyBlast finds a very fast way, realizes a very fast way to have an iterated alignment of profiles against sequences. And in fact, finding many, many hits. PsyBlast is the tool of the day. The tool of yesterday, I'm saying that in a sense last year, Clustal was the most used program in the world. I believe just now over the last 12 months, PsyBlast has taken over. Clustal W, Clustal X, is a dynamic programming. Again, it's a brute force, very well done. PsyBlast starts with a sequence and finds lines against the interdatabase. This one doesn't. What that one does is it starts with your PsyBlast run, and you go a little bit further, and its strength is to in fact align better whatever it found. Clustal. So first the naming convention, it started with Clustal V in '88, Clustal W, Clustal X, Clustal Omega, and that in fact comes on the website with a title, The Only Alignment Program You Will Ever Need. That's the latest Clustal Omega publication here from the group of Des Higgins, the complete name. The idea behind Clustal is, again, we start from the doodle approach. You do an all against all, find pairs. I'm sorry. Now the unique aspect really is in how Clustal handles these pairs. The way pairs are found is through phylogeny, through a tree. Let's begin with phylogeny. It's assume you have one protein, you have another protein that sort of evolves from this one here. It just gets a little bit longer. And we have another ancestor. It's essentially the same thing. Now we have another ancestor, and we already, in my logic that I applied before, sort of a situation with the second one. Maybe that already happened by duplicating some elements like two domains. So that means we now have a duplication, or in another version of that protein, we can have this light blue, meaning that actually does something slightly different. So suddenly we diverge in terms of function, structure. The original part here is still here, the original domain is still here. We move on. We in fact now duplicate the entire thing. So both domains in some sense. We move on, we slightly change this one. And we move on, and then suddenly, since we have the light blue, we have the dark blue, we can bring in some totally new element, the red one. So that's one assumption. Let's have another one. We have the green one, and we start from a longer one here. This time we cut one domain away, we evolve a new part, we duplicate, slightly evolve away, and in fact now in a similar logic have something very different. What we see from this tree that I made up here, maybe this. Okay? What we will assume from this is that in fact we have a two-domain protein, and we have no idea about its actual evolution. So this is the truth, and this is the fiction. We may go fully in the wrong direction. We cannot reproduce a tree. Sequences evolve way too fast. And we don't in fact have all the species that ever lived and their entire sequences. Even if we had, we may not be able to recognize the connections. There may be jumps in there. But we are far from having them all. that means that whatever tree we see is an assumption. And there are alternative trees that are possible. And that, in fact, is one of the modern ways of handling evolutionary trees. You compile the probability of getting one particular solution. Cluster uses one particular solution. And in this one particular solution, the unique way in which it does it is depending on exactly how similar A and B are, any pair, they decide what substitution matrix to use, what gap penalty to use, how to align them. So every pair alignment in that list is treated in sort of an individual way, with a lot of background information about how sequences behave. It clusters and does this. Again, cluster is fairly quoted here. Google Scholar numbers from May 2014, over 100,000 citations. Cluster W is 47,000 times, cluster X 30,000 times. The latest one, published 2 years ago, is just about to begin to grow. The people behind it, mostly Des Higgins, the original one, Toby Gibson. Toby Gibson is important because he really, I said initially that we want to align proteins with a computer, but some people do it with hand. Toby does it by hand or visually. He's great at that. And in fact, Des essentially programmed, there was a great team, these two here, programmed into cluster exactly what Toby said would be right. Toby had the biological intuition, and Des had the ability to mathematically put that into the computer program. Julie was the one who sort of put a nice graphical interface into this. This is a simplified version of the story, but there's a lot of truth to it. I believe that clearly cluster W, or the cluster suite of programs are very, very good. But there are others that are good. There are some others that are faster. What is unique is their visual interface. You can just make a nice, colorful plot and put it into a paper. And I believe at the end of the day, that's one of the important reasons why cluster is so successful. You have really 3 elements. It's unique in the world. There's no other example where 3 people have come together who have contributed these completely different ways of doing that. If you ever want to be successful in computational biology, and maybe in many, many other fields, try to find an alliance like that. For sequence alignment programs, there are hundreds, maybe thousands out there. There's one that realized that. But there's absolutely no reason for you not to be part of that one thing in the future that will change the world. But this is sort of one way to the secret of it. Des Higgins, over 100,000 publications, 94 in May 12, H-index of 52, here in Dublin. Toby Gibson, who is at the MBL, and Julie, who is in Strasbourg. Okay, another example. So Maxom, and I'm presenting that Maxom is sort of a program that is not really used anymore. These two are the leaders in the world. That is not, and I'm going to get into the next suite of programs that is sort of another coming up solution. Maxom is important because it has a different focus. It comes from Chris Sander and Reinhard Schneider. And the different focus, cluster really tries to align multiple sequences, and it has no notion of any one in this entire set of 100 sequences that are aligned being different. Maxom has a different view. Maxom says the first sequence is the one that is special to me. It's, say, the human protein that I'm studying. So I don't want an insertion in this one. This is my sequence. I want to know everything about it and not about its relatives. And that's an interesting philosophical perspective. BLAST does the same thing. It sort of aligns everything with respect to the first sequence. And that, in fact, creates something that is different. What is the type of multiple alignment that you want? One that sort of treats all sequences alike, or one that sort of enslaves everything to one sequence? And I believe that is the great aspect of Maxom, and that in some sense we should find a way to get back into the world. Now, Hammer, Sam, hidden Markov models. That's the next generation. Hidden Markov models got into biology originally through Anna Schroch, who visited in do I write it? Santa Cruz, University of California, Santa Cruz, and visited David Hausler, and David Hausler was a theoretical physicist, and interested in algorithms and machine learning and the like. And Anash, in fact, was one of the people who had introduced neural networks on a larger scale, had written a book about neural networks, and after this book about neural networks, he sort of began looking into hidden Markov models and thought that would be interesting to have hidden Markov models in biology. Went to David, and David is the type of person who's open to anything, and sort of sat there, yeah, cool, let's do it. Kevin had to do the math. Kevin is a little bit more of a grim guy, but he ultimately jumped into it. In parallel, Sean Eddy did something, also introduced hidden Markovs in England with Richard Durbin, but that also was when Anna Schrock had come back to Europe. So the way to use them, essentially, have you ever seen hidden Markov models before? I should have had a slide of hidden Markov models in the first. So those are the two sort of major themes of hidden Markov models here for the SAM, and these slides are in fact from SAM, from Kevin Karplas. Kevin Karplas here on his bike. That was stolen. So the way you do it, you start with a single sequence. Then you build a model, just like we did before, a profile. So you find everything that is similar to it. This model is, however, a hidden Markov model. A hidden Markov model, let me go back into this one here. You have a sequence and a probability, those are the hidden states, a probability of going either into an insertion state or into another sequence alignment state. And then you have a probability for every single alignment. And that is essentially the model. At every single position, you can either insert or align a particular residue, and then which residue. Essentially, you build a probability profile for that. And you can learn that from a bunch of sequences. Okay, that's the basic idea behind it. So you begin by in fact compiling the probability of observing a state A, and that in fact you get from the observation, the number of states that you have. It's minimal for a peak distribution, and is maximal for a uniform distribution, of course. Now, consider a particular residue position I before any amino acid is aligned. So any position here. What's the probability of aligning a particular residue there? So you don't have a model. The first step. How do you sort of initialize your model? You want to learn probabilities from observations. So I'm going to throw sequences at you that I tell you are related to that family, and you will use those to in fact learn. At every single position here, the X, that is not a letter, the F, the T, the N, so what is going to be aligned here? You're going to learn from cases. But before you learn, in the zeroth, here you begin. So what's the probability for aligning an F to the F?
Probably will initialize with a uniform distribution.
So you will do 1 in 20. Well, will you really do that? So first of all, it's funny that you say that because you like conservation. So first of all, you will most likely sort of overweight F, right? F is already better. So conserve position in some sense. You know, even for a starter, you may want to start with something that's one way of doing it, right? But in principle, you're right about that. Is there any simple improvement of that? So 1 in 20, and then sort of give F, give identity some other factor. Can you imagine any simple, fast improvement? So you would clearly not do that if, say, the F or only 19 residues would happen, and F would almost never happen, like the X. Like, let's just assume the X is amino acid 21, which in fact is selenocysteine, exists in eukaryotes, but it's very rare. But you would not give selenocysteine equal probability because it's almost never observed.
Could we use the distribution on the sequence that we know?
Yes, oh, that's an interesting one. That's an interesting one. Now we begin into, let me repeat what he said. We are going to use the probability of the distribution in that sequence that I'm looking at. And that clearly is the simplest thing you can do, because that is in your computer at the moment that you begin your model. Now, in fact, it is one way in which people have been doing that. The reason, or what I wanted to say here, computational biology is often about having alternatives and no clear idea how to choose which way to go, and that is one of the major complications in this field. That is one way in which you could do it. Another way in which you could do it is look up the database frequency. Now we have 3 possibilities, and ultimately, in the second, maybe equal probability is not good. Equal plus F could be as good as the other 2. Clearly, if your sequence is sort of biased, the idea that you use your sequence rather than database is a very good one. So it's not evident how to start. But we have now better starts than, I believe, we have fair enough starts. So background probabilities. The background probabilities would then, in this particular sequence, read out this way, where the height of the letter is proportional to the probability of, in fact, aligning that residue at that position. Okay? So the bits saved by aligning that position. So once you have the alignment, now you have a posterior probability. So if the position is conserved, then the information gain is essentially very little from aligning it. The bits saved are 0. If the position is completely varied, it's H0, some constant, and Hi minus H0, so the top that you can get by full conservation and the lowest that you can get by completely varied sequence are the bits saved, and in fact that is what I showed here. The slides come from Kevin Kaplas, by the way. Now we have another problem. All of this is good if I have a huge alignment. But some of the positions, in fact, may not have many sequences aligned. Again, sort of the domain protein here. Some of you may see this here. But in between, there may be residues where I have almost no alignment. And that means that I suddenly may have the indication that something is completely conserved only because there is nothing aligned there. That's a danger in this. In that one, then, in fact, if you do it right, you let the background dominate. And we are back to the point that, in fact, your choice of the background, of the initial state, is a crucial one. It may not very well be clear how to start, but suddenly, for these kind of situations, it becomes very crucial how you did that. So you have to look at alternatives and find a way to in fact, differentiate between what is better. The problem is, now you have a possible case, what is better means you train the model in some sense, and that is the problem that you can overtrain. Mind you, we are talking about one particular family here. Maybe what you do is true for that family, not for the next one. For the one where, in fact, the sequence bias is very high, not this one, but something that would have a lot of A's or something like that, it would have been better to start with the sequence itself. For another one, it would be better to start with the background from the database. Okay, so we build a model, use this model to find new ones. So this essentially corresponds to the profile I showed you. And this corresponds to the I in some sense, the iteration of PSI-BLAST. So now you have a profile, you align new sequences that you take from the database into that profile. Right? And then you do it again and again, and at the end you hope for convergence. That's the idea of SAM. Some issues are, again, what I already talked about is how we treat things that are completely conserved because there's overall not enough. Completely conserved, in fact, in the language of hidden Markov model is a multiplication of 0. That is not sort of a great thing. So there's a regularizer, so there's an artificial way. That is a problem that they had to deal with, but there are other problems. The hidden Markov model, I should have spent more time on explaining the hidden Markov model in the beginning. I'm sorry. But the hidden Markov model assumes that the state I only depends on the state before, I minus 1. Again, in the alignment, all the alignment methods that I talked about so far, we always assume that what I align at position I is completely independent of what I align at position j. I told you this is not biologically true, but this is the complexity that we barely can handle. Here, essentially the same thing is true. A hidden Markov model, I said, PSI-BLAST is the most frequently used method, followed by Clustal. Most likely, then comes Hammer, because it's used in a database called PFAM that is fairly frequently used. Let's go into some totally different area. T-coffee. T-coffee uses genetic algorithm. And again, any method that you can dream up has been used to align sequences. Those are just sort of the ones that made it. Now, T-coffee is one that made it because it ultimately was refined to produce very accurate results, although it takes a lot of time. The method comes from Cedric Notre Dame, developed in the group of Des Higgins with the help of Jaap Herringer. That's Cedric, now in CRG in Barcelona. If you look for a position in biology, this by all means is a nice place to be. So, you begin with a library of local, I'm sorry, I have a cramp coming up. You begin with a library of local and global alignments, and then you do a genetic algorithm. So a genetic algorithm essentially sort of grinds up all pairs and sort of randomly superposes them. That is this library, for instance, library. So essentially what I mean is pairwise alignments, okay? Some of them are local, some of them are global. Remember, global again, I force the alignment from one end to another, which in fact is exactly that issue that I want to, in many cases, align the entire plotting. You grind them up. Grind them up means you essentially keep only bits, okay? Every sort of, you hack them apart and you sort of reassemble them. And you build a genetic algorithm to essentially do all the reassemblies and assess these reassemblies according to their fitness. So that's the slide from Cedric. So you begin, for instance, with this one here, Garfield, the last fat cat, Garfield, the fast cat, Garfield, the very fast cat, the fat cat. You build these alignments by local pieces, and then you extend the library by bringing in new bits. You realign, you run the genetic algorithm. The beauty of the genetic algorithm, and this Cedric also likes tea, as you can see, or was it coffee? So the great thing of that one is you can import any kind of alignment, local alignment, multiple alignment, global alignment. You can input structural information. So what I said before, you can put in things where you know that the helices match. You could put in some expert knowledge, so the way, for instance, they are secreted or not. All of this could go into this middle. All of this information will help to drive the genetic algorithm to go into a certain direction in space. That certain direction in space will always have to do with the probability in which you sample the original states. The more you know, the more that will drive your final alignment, but still the final alignment will deviate from it because it will create new connections, as any genetic algorithm. So genetic algorithms do essentially re-pairing. And that ultimately produces a multiple alignment. Tea coffee is much, much slower than everything I showed you before. It requires pre-processing, but it allows you to bring in important additional information. SS Search and Psi Search are newer programs that are faster than Cluster. SS Search is similar to Maxom in the sense that it requires or it assumes a hierarchy. However, again, most of those are not very well used at the moment. Anything more fancy? Well, profile alignments. What would that be? Any idea? So sequence. Okay, let's go back to the question hour again. Sequence alignment. What does that mean? Is it too trivial, or are we in the sleeping part of the day? So essentially you use the information only from a pair of sequences. All you need is one sequence and another sequence, and you're not going to use anything else other than maybe background sequences, background information. But essentially for the alignment, all that drives you is what you have, those pair of sequences. Sequence profile means you take a sequence and align it into a profile. Profile, you compare two profiles. Like this. How would you do that? I take your answer away from you. You look only at the conserved positions, the 4 here, and compare those. But, well, not but. That's one way. But since I said that, that is no longer an option for your suggestion. Any other way? Types. I'm sorry, you have to help me out. I didn't get it.
Profile type.
File type.
Profile type.
Wild.
Profile type.
Profile type.
Yeah. You can divide the profiles into type and then compare them.
Try again. You're still not quite getting through in my head.
And divide the profiles into types and then compare.
But how do you divide profiles into types?
For comparison.
Yeah, but how? So, you want to cluster profiles.
Yes.
But that, you see, the point is, that's a good point. But ultimately what I'm asking here is how can you cluster? Because clustering is already a comparison. Clustering says you have two profiles that are or A is similar to B, but they're both dissimilar to C. There's one way of clustering. You find similarity in distance, right? Or only similarity would be enough to cluster. But my question here is, how do I find similarity? What is it that I look at, in fact, to compare A and B? You had it?
I was thinking about making a summary of the profile.
How do you make a summary of a profile?
For example, using probabilities for each position.
Yeah.
You say that this letter has this probability, this letter has this probability, so then now you have numbers.
Yes.
And with those numbers, you can then compare with the similarity.
Okay, so the statement is, I'm just putting myself on a row I. I compile the probability for every single 20 amino acids in row I. That gives me a 20 vector with probabilities. I can compare from this profile a row, and I get the 20 probabilities from this one. Now my question is, how do I compare these two?
There are standard methods.
Yes, you're totally right. So there are standard methods. Again, welcome to computational biology. The trouble is there are many standard methods in which you can compare two vectors. You can just multiply them, you can average them, you can sum them, you can... Which one is right? Big problem. The big problem underneath is that we move into a field where it is more and more difficult to get real life cases. Remember, in the last lecture we discussed how can we assess alignment accuracy. And I'm not sure whether the person who said that is in the room. He said here then, the point was maybe we can ask experts to give alignments to it, like the one from Toby Gibson. The problem is now we're trying to find similarities between things where the experts no longer see the similarity. So we're moving more and more in a direction where no expert can help us. That means that to test all these alternatives is really, really complicated. One hope, in fact we have too many free parameters, is the problem of the day. And in fact, here's another issue. In some cases, we want to possibly, in fact, remember correlations. So maybe what you see here is you go in this row from a positive to a charged residue to not a charged residue, from a charged to a not charged. Maybe this is a piece of information that you want to keep. You want to maybe keep that this is 3 times R, this is 3 times K, and 2 times W, and not the overall probability. Because it's very, very important that these two facts in this particular case, and this example is chosen such, that they are in fact correlated. They are in contact and there's a good reason for this correlated change. In the probability idea that you have, you completely throw that out. But again, so that is what people do. Yeah?
Question, isn't it random how they're...
No. So, again, the question here isn't the arrangement, so this is 1 protein, 2, 3, blah, 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 isn't the arrangement in which they come random? And let's get back to 2 different answers. Maxim SS search says the first is my sequence that I'm caring for. So immediately randomness is killed because from here on I write the second one is the most similar, second most similar, blah, blah, blah. Okay, that's an order here. Looking at cluster W, where everybody is treated alike, we begin also from pairs of most related things and build up whole alignment such that in fact this overall is most identical. The start in cluster is with a tree. The tree, I said, we don't know the tree, so there are many mistakes. In an extreme case, I could say the tree is random, but not quite. So saying that the trees that we see today from evolution contain a lot of errors and are problematic is not meaning they're completely random. So the trees are not random. In the cluster W case or cluster omega case, this would also imply some order. You can in fact at this point not imagine any example where you completely would randomize that. Again, ultimately when tea coffee may be the closest to random, but tea coffee in the end also grades. And it builds it onto something like Cedric originally came from DES, uses a similar tree idea. So no, it's not random. And in fact, in this particular case here, the non-random list would contain the important signal. You could even imagine, and there are other methods that do that, from my brain doesn't work today. Boris Sloan Kettering, but I don't know the family name of Boris. That in fact tries to rearrange this multiple alignment such that signals as the ones that I show you here come up. Okay, now that's another problem in profile. Here is a sheep version to profile alignments. The consensus, and that comes from Dariusz Przybylski, who is now at the Broad Institute. Profile sequence in terms of quality and CPU is somewhere here. It's fast, quality is not as good, and not as widely covering. Profile takes a lot of CPU, is very good. And somehow here's the idea to find something in between these two extremes. We begin, so here is the sequence aligned. Those are essentially the probabilities of that, the residue, what I see compared to background. And we now pick up the particular position, and star residues would be essentially the kind of residues that you said before are conserved. Although conserved is according to this substitution matrix. So compatible with the substitution matrix, what I see is like conserved in that substitution matrix, and then I write out these star residues. Okay? And then what I do is, when I come with my new sequence, I in fact don't align it against a profile. Instead I align it against a consensus sequence, which is just another sequence, and it means that I do something like just another pairwise alignment. Now I can do the same thing on a different level. I can do consensus against a profile. Doing consensus against a profile is almost like profile against profile, but it's much cheaper, because my consensus sequence is just a sequence. Now I align it against a profile, and I can do that with PsyBlast. I can use the program that I described to you, do the same thing, and have sort of a cheap consensus profile alignment that I use an existing program for. And when you do that, what this slide will tell you, if I gave you enough time, is that you get a very, very accurate alignment. You do much, much better than PsyBlast, by just using PsyBlast, simply replaced by, instead of sequence against profile, you do consensus sequence into a profile, which is a little bit like a profile against profile, but very, very fast. Just the same speed as PsyBlast, one iteration of BLAST. A similar idea here is used by H.H. Blitz. It's a modern work from Johann Söring, who's now at the Max Planck Institute in Göttingen. This is the publication here for it. And in fact, originally that was the development also done by Andrei Lupas in Tübingen. You start with a query, you do an HMM, you do a fast pre-filter, and then you do against the fast pre-filtered one. So fast pre-filter means you cut down your database from 55 million to 2 million or 3 million. For these 3 million, doing all against all. And this, in fact, is done with a very, very similar idea that I presented to you. So now we essentially do something like a consensus to profile alignment with an HMM. That speeds it up tremendously. The difference is that in the H.H. Blitz approach, this consensus is really a blow-up of the alphabet. Accepted HMMs, rejected HMMs, and that in fact is the only method that allows you currently to do something with an HMM that essentially does a profile-profile comparison on the level of almost the entire database. And that clearly seems to be the best method out that exists and finds relations in 3D structure that are very, very remote. Sequence, why bother to align sequences? Again, the background is we want to find homology, we want to find evolutionary relation, we want to find things that are first in the safe zone, twilight zone. So safe zone is pairwise BLAST, pairwise dynamic programming. Twilight zone is now you need profile, so sequence profile alignments. Lower part of the twilight zone is maybe profile profiles. Let me repeat some relations in sequence space. So the sequence space, structure space, this and that are different sequences. Different sequences fold into different structures. Similar sequences fold into similar structures or identical structures. Most sequences don't fold at all. Similar sequences, as I said, similar structure, similar sequences, typically similar function. This relation is not as clear, that's why this arrow is thicker here. Similar structure typically has similar function, and similar function very often has similar structure. Now, let's get to the final part of today. That's the midnight zone, or let's have questions the last 12 minutes. Whichever do you prefer? Midnight zone. From the twilight, was that? Sorry. From the twilight to the midnight zone. So again, safe, profile sequence, midnight zone. What's the midnight zone? The midnight zone is essentially trying to see relations between proteins for which I cannot recognize on their sequence their similarity. In order to identify those, I take proteins of known structure, pick out a sequence unique subset, do the sequence unique subset against all, and I'm essentially asking how many pairs of proteins do I find that in fact have a certain level of sequence identity and have similar structures again. So 100 to 0% sequence identity, and this is what I observe. Right? So everybody would be sort of happy with that. When I look at the data, I see more like that today because PDB is biased, so I see something that is more like this. And that, by the way, is the random background peak. But that is not true. That's not at all what I see. What I really see is this. That's the random background peak, and that's the true data. And now let me put, I should have had the background here. I'm sorry. So the way this graph looks here is different from the way it looks here. There are technical reasons. Essentially, this line here should be lower, much lower compared to that. So the basic message here is that I'm looking for all the pairs of proteins that have similar structure. And what you see is this. Meaning, compared again to random. This is random. I randomly superpose two sequences. Red. I find all things that have similar structure. Black. Very, very similar to each other. In particular, given the scale. In some sense, the random sequence identity is in the ballpark of 5%. 20 residues, 100% sequence identity, 100 by 20 is 5. That's roughly the 5. There's a slight deviation. That to some extent brings us back to the order and sequence and the like. This is what I observe for similar structure in the ballpark of 10. The difference between these two is the amount of space you have to create the diversity of all the folds. It's a tiny difference, right? And again, this is where we have most relations that we want to find. That's the midnight zone. So that's way below what we can find from sequence alignments. Not even profile alignments get us there. No, certainly not sequence. They stay here. Sequence profile goes somewhere in here, but nobody reaches here. That is the problem of the midnight zone. Cannot be reached. In some cases we can intrude, when we have very good profiles, but in most cases we cannot. What it seems to imply for evolution is that evolution may have had enough time to completely diverge, to try out all possibilities that there are. We cannot evolve further. So going back here, this is what we observe. This cannot hit the red. If it would hit the red, then overall random and signal would be identical. That cannot be. They must differ. So can the black, if we had another billion years, would the black move another percentage point below? Maybe. But this is remarkably close. We could argue that in fact the difference between black and red is 3 percentage points, is what you need to maintain function. And that's it. So you reached the end of the degree to which you can sort of explore this space of divergence. Anyway, it also means that convergent evolution, what I mean here, convergent to the same structure. So now we are so low. So divergent means you start from coming to the same sequence, diverge, maintain the structure. That's the idea of divergence coming from here. Convergence, you start from 0, which actually is not 0, for distribution is 5. And you see these two things are so close that you can in fact not distinguish in terms of numbers these two scenarios anymore. Again, we're not talking about convergent evolution as it usually is used in terms of functional evolution, but we're talking about convergence to the same structure. And for that, what many people believe is not happening very often cannot be distinguished from divergence. So it may have been a frequent event at this point. So in the next one, we get into secondary structure. Secondary structure can be used. Secondary structure can be used to align proteins, and that's the part that I wanted to skip there at the end. On Thursday, and next week, so this comparative modeling is going to be on Thursday. Comparative modeling, in fact, means that I take a sequence, I run against the database of known structures, I find relations, and I use those relations to predict 3D structure. Known structures means I know for every single residue where it sits in 3D, and I simply look that up. That's comparative modeling. More about that on Thursday. Thank you for your attention.